Hello, everyone, and welcome to this Genetic Engineering and Biotechnology News webinar. It's entitled Analysis of N-Glycan Released from Biopharmaceutical Glycoproteins Using HPAE-PAD. One of the most critical activities involved in the development of a novel biopharmaceutical involves protein glycosylation. Now, it's a fact that new drug discoveries get all the headlines, and all the benefits of FDA-approved biotherapeutics are touted in most forms of traditional and online media. But if the analysis of the glycan pattern of a recombinant protein drug is carried out incorrectly, that's a problem. And all the data regarding product characterization and quality control will be called into question. In other words, drug development here will be set back, and any headlines that may appear will definitely not be positive. How to avoid this? That's what today's webinar has been designed to address. Our panelists are going to discuss how a specific technique, HPAE-PAD, can be used to perform accurate and consistent mapping of N-glycans from biopharmaceuticals. Let's meet our panel. Peter Yandick is Principal Scientist in Research and Development at Thermo Fisher Scientific. Peter will provide an introduction to the analysis of carbohydrates using HPAE-PAD. Sebastian Kanzia is Head of Oligosaccharide Analytics at Glycothera. Sebastian will further illustrate why HPAE-PAD, which is also known as HPAE-PAD, is considered an optimum chromatographic method for accurate analysis of N-glycosylation. I'm John Sterling, Editor-in-Chief of GEN, and I'm going to serve as moderator. Please feel free to enlarge the slide images or download the complete presentation. At any time during the webinar, you can send in a question for our panelists. Type your question into the Ask a Question box on the lower left of your console and then hit Submit. The panel will try to answer as many as possible during the question and answer segment that takes place after all the presentations have been made. So if everyone's ready, let's get going. Peter Yandick will be our lead presenter. Peter?
Thank you, John. As already stated, we will be reviewing analysis of oligosaccharides by high-performance anion exchange chromatography with pulsed amperometric. We will start out with a quick look at what is high-performance anion exchange chromatography with PAD. There are two contributions making HPAE-PAD a powerful tool for carbohydrate analysis. First, there are the efficient resin-based anion exchange columns of a Carbopack series. The Carbopack columns are used in combination with pulsed amperometric detection in specially designed 3 electrode cells utilizing optimized sequences of potentials called waveforms. There are two key resins types in Carbopack columns. One of them is shown here. We see a schematic representation of a latex-based pellicular resin bead. The bead consists of 3 regions. Region 1, an inert, highly cross-linked, non-porous ethyl vinyl benzene divinyl benzene core. Since the bead is inert, it is ideally suited for use with hydroxide elements needed for carbohydrate separation and detection. High cross-linking imparts A, high mechanical stability for operation at high pressures, and B, excellent chemical stability for compatibility with HPLC solvents such as methanol and acetonitrile. Region 2, a thin 100 angstrom sulfonated layer on the surface of the ethyl vinyl benzene divinyl benzene core. The purpose of this layer is to provide a means of electrostatic attachment of the latex outer layer. Region 3, a latex anion exchange coating is attached to the sulfonated surface of the substrate bead. The latex layer is the active stationary phase where most of the analyte retention occurs. The latex layer carries the ion exchange groups, in this case quaternary ammonium groups. This layer is permanently attached to the support particle through electrostatic interactions of the quaternary ammonium groups in the latex with the sulfonated layer. I should say that all of these ion exchange materials can be operated in over a large pH range, typically at least from 0 to 14. This is not possible with silica-based reverse phase materials, which typically have a much narrower pH range Allow me to summarize the scope of applications of a Carbopec family of columns and the analyte classes that have been optimized for. To sum it up, a Carbopec column exhibits follicular design, fast mass transport, fast diffusion, high efficiency resolving power. Fast reequilibration after gradient runs, 10 minutes or less. High pH stability, as I already said, 0 to 14, comparable with hydroxide elements used for separation. High cross-linking, which imparts high mechanical stability. Maximum back pressure can be up to 4,000 or 5,000 psi in some cases. And solvent compatibility. Additionally, we have seen in past seminars that we also have capillary versions of these columns. They are Carbopec PA20 and Carbopec PA10 columns. For those of you who are new to amperometric, let me just quickly mention a few key points to get you up to speed. Amperometry measures current or change resulting from oxidation or reduction of an analyte at the surface of a working electrode. Typical working electrodes used are gold, platinum, silver, and glassy carbon. For carbohydrate analysis, gold is used as the electrode surface. Oxidation is when an electron goes from the analyte to the electrode, and reduction occurs when the electron goes from the electrode to the analyte. The resulting current produced is measured by the electrochemical detector. Carbohydrates are detected by measuring the current generated when they are oxidized at the surface of gold electrode. This slide shows the oxidation reaction. As the molecule is oxidized, it generates electrons. It is this charge that is picked up by the detector and is proportional to the concentration of a carbohydrate that is being oxidized at the electrode. The reaction generates hydrogen ions and is therefore driven by high pH. This makes PAD, pulsed amperometric detection, an ideal companion for HPAE, as the separations are done at high pH. They must be done at high pH. This detector does not oxidize all of the carbohydrate present. In fact, it only oxidized about 1% or maximum few percent of carbohydrates. It is therefore compatible with preparative chromatography and compatible with hyphenation techniques like mass spectrometry, since 90 to 99% of the material will be left intact. The electrochemical cell used for carbohydrate analysis is the so-called free electrode design. It consists of, first, a working electrode, and working electrode is where the analyte is oxidized or reduced. Current through this electrode is measured by the electronics of the detector. For carbohydrate analysis, the working electrode is gold. Reference electrode sets the potential of the working electrode. For the analytical format, a silver chloride combined reference electrode is used, and for the capillary format, we like to utilize palladium hydrogen solid-state reference electrode. Counter electrode prevents current generated at the working electrode from passing through the reference electrode, which would otherwise cause chemical changes inside the reference electrode. Here we have a photograph of different types of working electrodes. These are available either in conventional format, where the gold is embedded in a CalF block, for example, or in a disposable format. The disposable electrodes are microfabricated as follows. The polymeric film is cleaned and dried. After assembling it with a stainless steel mask, we transfer it into a sputtering chamber. The chamber is then pumped down to a high vacuum. With argon plasma on, we start to sputter on a titanium layer. In the next step, we sputter gold. The opening in the mask defines the sputter pattern, the shape of the electrode, contact lead, and contact pad, which is this big round thing at the bottom. Here you can see the various formats and types available. Why disposable electrodes? Why did we develop them? They can be manufactured with a very high reproducibility and efficiency. They are cost-effective, especially in quality control and also in any high-throughput environment. They are very easy to install with fast equilibration, which is minimal delay after installation, and exhibit an excellent electrode-to-electrode reproducibility. have a predictable stability of response and no conditioning or polishing is required. The free electrode cell, consisting of a working electrode, a reference electrode, and counter electrode, is connected to the outlet of a suitable carbopec column. Whenever a separated zone of a carbohydrate reaches the detector cell, the working electrode is generating the current measured between the working electrode and counter electrode. The current is electronically processed by a detector. The recording of the current over time is giving rise to peaks positioned on a baseline. The current is at a baseline level when there is no carbohydrate passing through the cell. The peak area of the resulting peaks is proportional to the carbohydrate's concentration in the zones separated by the carbopec column. There are three key modes of amperometry, or the measurement of electrode currents. These are DC, or direct current. In this mode, we apply a single potential and measure the current generated from the oxidation or reduction process. The next mode is pulsed electrochemical techniques. In pulsed mode, we apply a series of potentials to the working electrode. PAD, or pulsed amperometric detection, it measures the current at one potential and integrates to get a charge in nanocoulombs. IPAD, or integrated pulsed amperometric detection, measures current at more than one potential and also integrates to get the charge in nanocoulomb. For potential waveform, we have mentioned waveform already. Waveform is a series of optimized potentials that are used for the detection. Pulsed amperometric detection brings about the ability to use these waveforms. This slide shows the preferred potential waveform for the sensitive and selective detection of carbohydrates. After the initial potential is established, the resulting current is integrated over a short period of time. Then the electrode is stepped to a potential which cleans it, first to a negative and then to a positive potential. Now the electrode is ready to be stepped back to the potential required for carbohydrate oxidation. This multiple waveform operation allows sensitive and selective detection of carbohydrates while keeping the electrode clean and activated throughout the chromatographic run. Comparison of chromatographic techniques for the analysis of hydrolyzed glucose syrup illustrates the remarkable resolving power of HPAE-PAD technique. Elution order is reversed with carbopec PA1 column as compared with conventional metal-loaded cation exchange columns. We are observing a much higher sensitivity by few orders of magnitude. As a transition to the following presentation given by Sebastian, I would like to mention the commercial availability of oligosaccharide standard from Dionex products from Thermo Fisher. It's a Q-seed standard of sialated N-linked alditols. This standard has a high utility for someone who separates complex oligosaccharide mixtures. This could be used for system validation, for example, when one gets unexpected results or when something is obviously not functioning. The injections of these standards can be used to verify that the system has been repaired or the system is working normally. And the good thing is that there is always a supply of this standard, which is relatively complex, that can be used in troubleshooting and for system validation. In summary, the great utility of HPAE-PAD originates from two unique contributions. First, it is a carbopec series of columns delivering separations in practically all classes of carbohydrates. All separations with carbohydrate columns are carried out in alkaline solutions. The alkaline elements are a great match for the second component of HPAE-PAD, the pulsed amperometric detection. The pulsed amperometric detection, or PAD, uses optimized waveforms developed for gold electrodes, and they enable highly specific detection of carbohydrates without any derivatization. This slide concludes the first part of today's webinar. And now back to you, John.
Peter, thank you for that very detailed presentation and for providing an excellent introduction to the analysis of carbohydrates using HPAE-PAD. You've perfectly set the stage for the presentation to follow, so thank you very much. If you're just now joining our webinar, welcome. As I mentioned earlier, we will be conducting a Q&A segment following the panelists' presentations. Please type your question for any or all of our panelists into the Ask a Question box on the lower left of your console, and then hit Submit. Our second panelist, Sebastian Kanzia, will now begin his presentation. Sebastian, the floor is all yours.
Thank you, John, for the kind introduction. Today I will talk about N-glycosylation quality of recombinant biopharmaceutical glycoproteins. But first, I would like to give a short introduction of Glycosera. Glycosera is a biotech company located in Hanover, Germany, and we are closely working with our industrial partners, which are national and international biotech and pharmaceutical companies, ranging from small startups to big corporations, and the development of human biopharmaceuticals, which are biosimilar, biobetter, as well as innovator products. We are GMP certified for quality testing of biopharmaceutical products according to ICH and EU guidelines. We have a strong background in quality dossier submission to regulatory authorities, and we were involved in the development of a number of biosimilar biopharmaceuticals, leading to successful marketing authorization. Based on the strong expertise of Glycosera scientific and regulatory management, we are offering the highest standards of reports and support in the preparation of submission files. This is a short extract of glycosylated biopharmaceuticals that have been analyzed in our laboratories. The variety of N-glycan structures to be analyzed range from highly sialated, highly antennary structures, which, for example, can be found in erythropoietin and represent about 40% of the molecular weight of the total glycoprotein, to truncated neutral N-glycans, which are typically present in recombinant antibodies and correspond to approximately 2% of the molecular weight of the entire antibody. In the subsequent slides, I will show N-glycan mapping procedures for biopharmaceutical glycoproteins for human use, which are on the way to the market, which have to follow the regulatory pathway for marketing authorization with the regulator by the EMA, the European Medicines Agency, and the European Union, and where human biopharmaceutical products have to be proved regarding quality, safety, and efficiency. On the other hand, we have complex glycoprotein structures due to complex biological manufacturing processes, including the producer cell line, upstream and downstream processing, where especially the glycan part shows a lot of different high complex structures that are responsible for many biological roles, which impacts the entire glycoprotein, like stability and protein folding. In order to fulfill the regulatory issues regarding quality, safety, and efficiency, N-glycan mapping procedures are required that are highly resolving, reliable, robust, and sensitive. Before we start with analytical data, let's have a brief look at an N-glycan structure. This presented N-glycan structure consists of a common core containing 3 mannose and 2 glucnec units. We have 2 fucosylation sites, 1 proximal alpha-1,6 linked fucose and 1 peripheral alpha-1,3 linked fucose, also known as Lewis-type fucosylation. Our N-glycan has 2 antennae. We are talking about a diantennary N-glycan. Each antenna consists of a gal-beta-1,4 glucnec unit. This antenna motif appears here repeatedly. We are talking about an N-acetylactosamine repeat. Our N-glycan has 1 terminal link sialic acid, in this case N-acetyluramic acid. Alternatively, the sialic acid and glycosyluramic acid, which is not synthesized in humans, could also be linked. The linkage can be in alpha to 3 or alpha to 6 direction. We are talking about a monosaccharide N-glycan. The other antenna contains a terminal gal alpha-1,3 gal motif, a glycan motif that is very common in glycoproteins deriving from hamster or mouse cell lines, and a glycan motif that is not found in humans. In the subsequent slides, the N-glycan structures will be presented in the simplified cartoon-like figure. These cartoon-like glycan structures were drawn by using the glycan builder software. I would like to demonstrate the immense variations of N-glycan structures on this diantennary complex type structure. Please be focused on the lower gal-beta-1,4 glucnec antenna of this diantennary structure. These are some potential modifications of one single antenna depending on the chosen producer cell line. Some of these glycan modifications are unwanted or potential antigenic glycan motifs, and for regulatory safety issues, we are screening for N-glycans from human biopharmaceuticals for unwanted or potential antigenic glycan structures. Among those are oligomonocytic glycan change, which occur in neutral and phosphorylated form, glycans containing the gal alpha-1,3 gal motif, glycans with peripheral Foucault-Lewis type motifs, type 1 structures, and derivatives of sialic acids like O-acetylated N-acetyluramic acid and N-glycoluramic acid. This scheme shows the workflow for primary structural analysis of N-glycans for dossier submission. With the scheme, I want to demonstrate the extent of sample preparation and analysis that need to be performed for such a primary structural analysis, which is very time and cost-consuming and not suitable for daily routine analysis of N-glycan structures. Routine N-glycan analysis will be performed according to the following simplified workflow. N-glycans were enzymatically released from the glycoprotein by PNGSF, followed by purification steps. For N-glycan analysis, we are using two different chromatographic techniques, HPAE-PAD of native N-glycans and normal phase HPLC of fluorescence-labeled N-glycans. Additionally, the N-glycans were analyzed via different MS methods, Molotov and electrospray. These analyses were supported by specific exoglycosidase digestions in order to identify specific glycan motifs. Very important is the use of complementary methods for reliable assignment of glycan structures. Today I will focus on N-glycan analysis via HPAE-PAD. For HPAE-PAD analysis of N-glycans, we are using a high-resolution Carbopac PA200 column, a gradient consisting of sodium hydroxide and sodium acetate, and for sensitive detection of N-glycans, a PAD detector with a 3 mm gold working electrode. Very important, there is no derivatization of N-glycans required. At Glycothera, we are using GMP-certified HPAE-PAD mapping techniques, and we are able to quantify N-glycan structures by applying a glycan library we established with more than 250 N-glycan structures. An example for a biopharmaceutical glycoprotein is erythropoietin, which contains 3 N-glycosylation sites with predominantly large glycan molecules, tetrasylated tetraantennary with up to 4 N-acetylactosamine repeats. This figure shows a tetrasylated tetraantennary structure and acetylactosamine repeats and proximal alpha-1,6 Foucaults. This is a representative HPAE-PAD profile of native N-glycans from an EPO biosimilar product which derives from CHO cells. The chromatogram can be separated in glycan groups with a different number of terminal linked sialic acids, starting with glycan structures without terminal linked sialic acids with 1 to 5 sialic acids, and the glycan group with 5 sialic acids sulfated N-glycans can be found. The N-glycan structures were identified using appropriate reference standards. Starting with the main glycan group, the tetrasylated N-glycans, the following glycan structures were identified. The main signal peak correspond to tetrasylated tetraantennary with proximal Foucaults. Then we find tetrasylated tetraantennary N-glycans with 1 LACNAC repeat. 2 isomer structures were detected with 2 and with 3 LACNAC repeats. In the trisylated glycan group, 2 isomer structures of the triantennary N-glycan were found. The 2,4 and the 2,6 triantennary structure, and triantennary N-glycans with up to 2 LACNAC repeats were detected, as well as tetraantennary N-glycan structures without and with LACNAC repeats. In the diacylated glycan group, the main glycan structure, diacylated diantennary, was identified, and also in the monocylated glycan group, the main glycan structure, monocylated diantennary, was detected. With one single HPAE-PAD run, most of the N-glycan structures were identified using appropriate reference standards. But for regulatory safety issues, as it's required for EMA submission, we perform a primary structure analysis, and we have to screen the sample for unwanted potential antigenic glycan structures. And this is most challenging in the neutral glycan group, as these glycans usually represent below 0.5% of total N-glycans from EPO. But before we screen for unwanted potential antigenic structures, we perform a previous preparative anion exchange chromatography run of large amounts of N-glycans using a mono-Q ion exchange column in order to receive sufficient amount of N-glycans for reliable HPAE-PAD analysis. This is the HPC-PAD profile of the neutral glycan group after mono-Q separation. And we found the diantennary N-glycan, oligomonocytic glycan change with 4 to 9 mannose units, and diantennary N-glycans with 1 and 2 gal alpha-1,3 gal motifs. Another biopharmaceutical glycoprotein is follicle-stimulating hormone, FSH, which consists of an alpha and a beta subunit, each containing 2 N-glycosylation sites. The alpha subunit contains predominantly N-glycans without proximal fucose, whereas the N-glycans in the beta subunit are predominantly proximal fucosylated. This is a representative HPC-PAD profile of the native N-glycans from FSH market product gonal F. Again, the chromatogram can be separated in glycan groups with a different number of terminal linked sialic acids, starting with the glycan structures without terminal linked sialic acids with 1 to 5 sialic acids and sulfated glycans. The N-glycan structures were identified using appropriate reference standards. Starting with the monosialilated glycan group, we see the diantennary N-glycans with and without proximal fucose. In the main glycan group, the disialated N-glycans, the diantennary N-glycans with and without proximal fucose were identified, as well as diantennary N-glycans containing peripheral fucose-Lewis type motifs, which represent about 2% of total N-glycans. In the trisilated glycan group, the 2 isomers of triantennary with and without proximal fucose were found, as well as phosphorylated oligomonocytic glycan change with 5 and 6 mannose residues. In the tetrasilated glycan groups, the tetraantennary N-glycans with and without proximal fucose were detected. Again, with one single HPC-PAD run, most of the glycan structures were identified using appropriate reference standards. For screening of unwanted potential antigenic structures within the neutral glycan group, which correspond to about 1.5% of total N-glycans, we used a different strategy. This time, the neutral glycan structures were collected during the analytical HPC-PAD analysis using a carbohydrate membrane dissolter device, followed by fluorescence 2AB labeling and normal phase HPLC with fluorescence detection. This is the normal phase HPLC profile of 2AB labeled N-glycans from the neutral glycan group, where we found the diantennary N-glycans with and without proximal fucose as the main glycan structures. We could identify diantennary N-glycans with 1 Lacnac repeat with and without proximal fucose and truncated N-glycans from the main structure, missing 1 beta-galactose residue where both isomers were baseline separated, lacking 1 antenna, also baseline separation of both isomers, and without 2 beta-galactose residues were detected. As normal phase HPLC of fluorescence-labeled N-glycans is a widely used technique for N-glycan mapping, I want to mention some important key facts. First, I want to go very quickly through the labeling procedure. The N-glycan will be labeled at the reducing end, where we find glucanac and ring and open chain form. Adding a fluorescence label like 2AB, the aldehyde group of the glucanac and the amino group of the 2AB label form a shift base, which is reduced to receive a stable linkage between N-glycan and fluorescence label. These are the labeling conditions according to publication from Big et al. From 1995, which were widely used in many publications and also in commercially available labeling kits. As the N-glycans were incubated in acetic acid at elevated temperatures, it has to be demonstrated that the N-glycans were not further modified during the labeling procedure, and one important issue is the desilation of sialated N-glycans during labeling procedure. For this demonstration, we choose a tetrasialated tetraantennary N-glycan structure with 2 Lacnac repeats before and after 2AB labeling, followed by HPC-PAD analysis. So in the upper HPC-PAD profile, we see the tetrasialated tetraantennary N-glycan before 2AB labeling. In the lowest HPC-PAD profile, we see non-carbohydrate signals from a mock 2AB labeling preparation, which were detected by the PAD detector, but were not detectable by the fluorescence detector. The second profile from above is a profile of the tetracylated tetraentyl N-glycan after 2AB labeling at standard conditions. What we see is the remaining starting material without label about 20%, and the 2AB labeled glycan structure about 50%. Additionally, the 2AB labeled glycans missing 1 and 2 sialic acid residues were also detected, representing about 25% of total carbohydrate signals. Further signal peaks were assigned as a reduced starting material and the starting material lacking 1 sialic acid residue. Desialation of sialated N-glycans during 2AB labeling procedure can be reduced by decreasing incubation temperature to 37 degrees and extending incubation time to overnight. But we just received 30% of the final product, the 2AB labeled N-glycan, whereas 70% of the N-glycan remain unlabeled. In many protocols for fluorescence labeling of sialated N-glycans, the issue of desialation is considered as neglectable, as in most of these cases, desialation was demonstrated with monosialated N-glycans. We also analyzed a monosialated N-glycan before and after 2AB labeling via HPAE-PAD. This is the HPAE-PAD profile of the monosialated N-glycan before 2AB labeling, and below is the profile of the 2AB labeled N-glycan using standard conditions. We see that we receive more than 90% of the 2AB labeled N-glycan and only minor signal peaks for the reduced unlabeled glycan structure and of the 2AB labeled structure missing 1 sialic acid residue, which represent about 4% of total signal peaks. We at Glycosera are using normal phase HPLC of 2AB labeled N-glycans as a complementary method for the analysis of neutral glycan structures. Compared to HPAE-PAD, we receive a 50 times higher sensitivity. We can go down to 5 to 10 femtomole, depending on the detector and detector settings, and we can get an enhanced resolution for truncated neutral glycan structures that can be found in antibodies. Typically, antibodies have one N-glycosylation site in the FC fragment with predominantly neutral truncated glycan structures. But there are also some exceptions, and one of them is cetuximab, sold under the trade name Erbitux, which has another N-glycosylation site at the FAB fragment containing predominantly sialated complex type structures. These glycan structures are therefore analyzed via HPAE-PAD. This is a representative HPAE-PAD profile of the native N-glycans from market product Erbitux. The chromatogram can be separated in glycan groups of neutral monosialated and disialated N-glycans. The N-glycan structures were identified using appropriate reference standards. In the neutral glycan group, we see diantennary N-glycans without 2 beta-galactose residues, oligomonosidic MAN5 glucanate-2 glycan change, diantennary N-glycans without 1 beta-galactose residue, both isomers were detected, and diantennary N-glycans. These N-glycans are typical glycan structures from the FC fragment. The following glycan structures mainly derive from the FAB fragment. These are the diantennary N-glycan with 1 and 2 gal alpha-1 3 gal motifs, triantennary glycans with 2 and 3 gal alpha-1 3 gal motifs. Within the monosialated and disialated glycan groups, which contain N-glycans with terminal linked N-glycolyluronic acid exclusively, the monosialated diantennary N-glycan with 1 terminal gal alpha-1 3 gal motif and the disialated diantennary N-glycans were found. N-glycans containing the gal alpha-1 3 gal motif represent about 30% of total N-glycans in Erbitux, and sialated N-glycans with terminal N-glycolyluronic acid residues also correspond to about 30% of total N-glycans in Erbitux. At the end, I would like to give a short summary on the just presented HPAE-PAD mapping techniques. The HPAE-PAD system is the most effective chromatographic system for N-glycosylation analysis of biopharmaceutical glycoproteins, with high resolution, even separation of isomer structures possible, and sensitive detection of N-glycans. It is a robust and consistent system, analyzing more than 100 samples per week. the quantification of neutral and charged glycan structures without prior derivatization is possible. It can be used for different applications like monosaccharide or amino acid analysis. Together with complementary methods like normal phase HPLC or fluorescence labeled N-glycans and MS analysis, HPAE-PAD meet regulatory criteria regarding quality, safety, and efficiency issues of N-glycans from human biopharmaceutical products. We at Glycosera have established a number of GMP-validated HPAE-PAD methods according to ICH EU guidelines for release testing of human biopharmaceuticals, which are also part of European Pharmacopoeia. I would like to thank for your attention, and I'm giving back to you, John. Thank you.
Well, thank you, Sebastian, and thank you again, Peter. You've both done a wonderful job in showing how HPAE-PAD can be used to carry out accurate and consistent mapping of N-glycans from biopharmaceuticals. Indeed, you've elicited a number of excellent questions from the audience for our Q&A session. Before we begin, I'd like to ask you to disable your pop-up blockers because a short survey on this webinar will be appearing very soon. We would very much appreciate your feedback on this webinar, as your thoughts will help us to continue to bring you topical and timely webinars in the future.
Okay, so let's take our first question from the audience, and it's for Peter Yandick. Can I use Hilic columns with pulsed amperometric detection?
Thank you, John. Hilic columns are typically used with mildly acidic eluents, also containing a relatively high concentration of organic solvents. Those two conditions are unfortunately not suitable for pulsed amperometric detection.
Okay, and Peter, we have another question. How long does a typical disposable electrode last when used for carbohydrate analysis?
We have two types of disposable electrodes, gold disposable electrodes currently. One of them is on a polyester substrate and the other on a Teflon substrate. The Teflon substrate electrodes who have been introduced recently last 4 weeks or longer. The polyester-based gold electrode, disposable gold electrodes, are specified to be used for 2 weeks.
And what method do you use to QC disposable electrodes?
Disposable electrodes are QC'd by injecting a mixture of 6 monosaccharides, and we specify for quality of baseline, noise, and also the response, minimum response for one of the monosaccharides in the QC testing. Each set of electrodes, as it is shipped, includes a quality assurance record, a sheet that lists all those data that were determined during the QC testing.
Thank you very much. And Sebastian, we have some questions for you. What amount of N-glycans are you injecting for the HPAE-PAD analysis?
Thank you, John. Usually we're injecting about 100 to 400 picomoles, but this depends on mainly 2 aspects. The quality of your system, your HPAE-PAD system, and the consumables. That means when you're using a 3 millimeter gold electrode, that's new, and on your glycan sample. So for instance, if you have a simple mixture of N-glycans, which are present in some antibody samples consisting of 10 to 15 different glycan structures, with one main glycan structure representing more than 80% of total N-glycans, we inject about 100 to 150 picomole of this N-glycan mixture in order to ensure that the main structure is within the linearity. But if you have a very complex glycan mixture, for example, that can be found in Erbitax, which I just showed, which has N-glycans from FC and FIB fragment, we inject about 300 to 400 picomole, as these Erbitax sample has more than 60 different structures, what we found here at Glycosera.
Okay. And Sebastian, what is the limit of quantification for N-glycan mapping via HPAE-PAD?
Here again, it's On one hand, it's the matter of the HPAE-PAD system. If it's in a good condition with a new 3 mm gold electrode and a Carbopack PA200 column to get a good signal-to-noise ratio. And on the other hand, it depends on the N-glycan structure, as large structures, high antennary, highly sialated, give a high response, whereas a small truncated structure gives a small low response. If you inject the same amounts, though the LQ for HPAE-PAD is around 200 to 500 femtomole, depending on the glycan structure.
And are you using conventional or disposable gold electrodes?
For carbohydrate analysis, we are using conventional electrodes, as our HPAE-PAD systems are running all the time, day and night. So for us, it's these conventional electrodes are more stable for our applications. And also we are using the 3 mm gold electrode, though we just have to polish the electrode every 3 to 6 months with sandpaper. But for amino acid analysis, we are using disposable electrodes, which are very convenient and they're ready to use. We just have to build them in and can start the analysis. And amino acid analysis, it's like an on-demand analysis at Glycothera.
Okay, Sebastian, how long does it take to set up an HPAE-PAD system for N-glycan analysis?
I would guess it takes about 3 to 6 months. It depends on the background, the equipment of the laboratory. But if you have one responsible person who can set up all the system to get the samples running compared to reference standards, 3 to 6 months should be the time you need for the setup of the system.
Okay, and where do you get your oligosaccharide reference standards from?
Well, on one hand, we prepare them on our own from different glycoproteins. We are purifying them after PNGA digestion using different chromatographic steps. But also we get them commercially. There's a company called Proteins from Portugal, where they have a huge selection of glycan reference standards, though that are our sources.
And Sebastian, how do you release terminal-linked sialic acids from sialated N-glycans?
Usually we use two different methods. One is mild acid hydrolysis, and the other one is enzymatically with sialidase. We prefer the enzymatic digestion, as with the mild acid hydrolysis. We see that we lose some proximal fucose and also terminal beta-galactose. So to not get any artifacts, we prefer the sialidase digestion for desialation of sialated N-glycans.
Very good. Okay. Now, how do you distinguish between sialated N-glycans containing N-acetylnuramic acid and N-glyconuramic acid?
These two different sialic acids, they have different retention characteristics on the PA200 column. So glycan structures containing N-acetylnuramic acid, they elute earlier than the structures containing N-glyconuramic acid, and for both we have reference standards. So by checking the retention time, we can identify the two distinct sialic acids on the N-glycans, but also after desialation, the released sialic acids can be analyzed, what we also perform, and especially the glycoproteins from CHO cells, which contain a low degree of N-glyconuramic acid. It's an important safety issue for analysis, as N-glyconuramic acid is not synthesized in humans.
And Sebastian, what is the lifetime of the Carbopack columns that you're using?
The Carbopack PA200 columns we're using, the lifetime, it's 3 to 5 years. It depends, I guess, on the batch of the column, but also on the samples, if they're purified or not, and of the number of runs. But I think 3 to 5 years should be the lifetime.
And another question, Sebastian, why don't you analyze the N-glycans via mass spec?
Mass spec? Okay, we also have a lot of mass spec instruments here, so we also analyze them via mass spec. As I said, we are using complementary methods for reliable assignments of glycan structures. And for example, one glycan motif that we have to analyze via mass spectrometry is the O-acetylation, as O-acetylated residues will get lost at high pH, what we are using for HPAE-PAD analysis. So this glycan motif we have to analyze via electrospray mass spectrometry.
Okay, and I have another question that either Peter or Sebastian can answer. You also mentioned that you can reach femtomole level sensitivity. What sensitivity do you get from 2AB labeling protocol? Either one of you gentlemen, or both?
Okay, what we observed for 2AB labeling, for 2AB labeled N-glycans, we are around 5 to 10 femtomole. That's the limit of quantification.
Anything to add, Peter?
Yeah.
I would basically confirm what Sebastian is saying.
Okay. So
femtomole range.
Okay. We have, let's see, 2 and 8. Yeah. Well, it looks like we've run out of time. So please note that this webinar will be archived for 6 months on our website, www.genengnews.com. If you miss parts of it, you can watch it again or you can recommend it to your colleagues and friends. I want to say a big thanks again to the panel for the outstanding presentations, and I want to say thank you to our audience for your attention and for your very thoughtful questions about various topics brought up during the webinar. And thank you to our sponsor, Thermo Fisher Scientific, who made this event possible. Bye for now.